Welcome to AP Biology. I'm Gail Fizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. Today, the topic we are covering is one that colleges have asked that all AP students study, even if it will not be on the exam. Colleges are supporting AP students this year by allowing this more flexible approach to the exam, where it will focus on a lesser amount of content than in other years. But in turn, they have asked that AP students learn today's material. To make this relevant to your upcoming exam, we'll also include some exam prep in today's lesson. Soon, after we've covered some of these key outstanding topics, we'll be posting the full schedule of review topics for the remainder of this course so that you can choose which topics you want to prioritize for review, and thus which classes you'll want to attend. Now for an overview of our lesson. So today we're going to be talking about topic 7.4, which is population genetics. I'll be introducing you to population genetics, science practice skill 3B, which is questions and methods. And we're going to be stating the null or alternative hypotheses and predicting the results of an experiment. You should already be familiar with the causes of natural selection, how natural selection affects populations, the importance of phenotypic variation in a population, and explaining biological concepts, processes, and phenomena. By the end of this lesson, you'll also be able to explain how random occurrences affect the genetic makeup of a population, describe the role of random processes in the evolution of specific populations, and describe the change in the genetic makeup of a population over time. So now for our warm-up. If you've been watching, you know that the homework from the previous night will be the warm-up for the next lesson. So this is from the homework from 7.3. Let's take a look at our data table and see what this is about first. Okay, incidence of antibiotic resistant in patients admitted to ICU from 1994 to 2000. Okay, we have a list of antibiotics, ciprofloxacin, gentamicin, ampicillin, imipenem, tazobactam, and we have the percent of all samples demonstrating antibiotic resistance in 2000, and then an overall change in percent resistance to antibiotics from 1994 to 2000. Okay, let's read the story. To determine the likelihood that a patient with a bacterial infection who is in the intensive care unit, the ICU, of a hospital could be successfully treated with antibiotic therapies, researchers investigated the frequency of antibiotic resistance in bacteria isolated from patients in ICUs in the United States from 1994 to 2000. The data from the study are shown in the table, along with the overall change in the frequency of bacterial resistance in patient samples from 1994 to 2000. Okay, what is our job to do? A patient was admitted to the ICU in December 2000 with an uncharacterized bacterial infection. Based on the data from the study, identify the following. The antibiotic that was most likely to be effective for treating the infection, and then identify, two identifies here, the antibiotic that was least likely to be effective for treating the infection. I'll give you about 20 seconds to do that if you are just joining us for the first time. Okay, I'll give you 10 more seconds. Okay, let's see what the scoring guideline indicated. All right, so there's 2 points here because you identified two medications, two antibiotics. And when you identify, it can be as simple as saying the word, okay? So if you put imipenem is the most effective and ampicillin is the least effective, you got both of those points. Okay, I'm going to go through this for a few more times in this amount of detail, and then probably by the time we start reviewing, I'll start dropping this, or then just narrow it down to what topic we're going to be reviewing. Okay, so the topic of tonight, we're doing population genetics, and that falls under the unit evolution or natural selection. And each topic has questions called learning objectives, and this is what we need to be able to do. And we need to know content in order to answer these questions. That content is called essential knowledge. Each topic also has an associated science skill, and this is how we're going to show that we know the answer to the question, the learning objective. The science skill that is associated with population genetics is questions and methods, and this includes a null hypothesis or an alternate hypothesis and predicting the results of the experiment. Today, we'll topic skill pair population genetics with questions and methods. Okay, here's what you need to know. All right, so the learning objective, explain how random occurrences affect the genetic makeup of a population. We're going to learn about mutations, genetic drift, two types. We're going to talk about bottleneck, and we're going to talk about founder effect, and then we're going to talk about gene flow. The first occurrence that we're going to talk about is mutations. Mutations are changes in DNA sequences. They can happen spontaneously in replication and result from environmental factors such as radiation or chemicals. Mutations can result in a recessive disorder or a dominant disorder. A change in a DNA sequence caused by mutations may affect the structure and function of proteins. Mutations result in genetic variation, which provides phenotypes on which natural selection acts. This changes the genetic makeup of a population over time. The next topic I want to talk about is genetic drift. And we're going to talk about two examples of that. We're going to do bottleneck and founder effect in just a moment. All right, so genetic drift is a non-selective process occurring in small populations. The smaller the number of organisms, the more likely the representation of an allele will change or disappear. There are two types of genetic drift that I'm going to talk about, bottleneck and founder effect. Okay, so for bottleneck, we're going to talk about the great prairie chicken. And I'm going to tell you the story. I read some research articles to pull all this together. In the mid to late 1800s, in the state of Illinois, there were millions of great prairie chickens. In 1993, there were only 50. The great prairie chickens lost natural habitats of prairie lands because of human activity, such as creating farmland. This reduced population had lost genetic diversity and variation. The genetic diversity of the great prairie chickens had declined over the years to this bottleneck. To prove there had been a decrease in the number of alleles, researchers used the DNA of great prairie chicken specimens from a museum as a control for the pre-bottleneck alleles. They extracted DNA from museum specimens and chickens from the central Illinois and other surrounding areas. They analyzed the DNA from the samples taken from the feather root, and they looked at 6 loci for differences in the number of alleles. To do this, they ran PCR and gel electrophoresis. Their results indicated there were 5 missing alleles. So genetic diversity had in fact decreased during human activity. Okay. Here is the state of Illinois, and this is not to scale, and these great prairie chickens do not represent any number. This is just a representation of there was a lot of prairie land for them, and there are a lot of great prairie chickens, and then that was reduced, and they were down to just 50 in 1993 of great prairie chickens. So if I said some words that you didn't understand, or you were thinking about 2 alleles, I added this part to help you out. Okay, so here we have a homologous pair. I'm going to get my mouse so I don't know if you can see that. No, I think I better do it with my finger. Okay, so here's a homologous pair of chromosomes. So this is, one came from your mom and one came from your dad. And so here is these dark bands. Those are actually genes, and they have a location, and we're going to call that a loci. And so if you, it's just like living on your street, okay? If this was your street, then you would have an address on that street, and that would be your location, right? So that's the loci that we're talking about. So you can have more than 2 alleles, and they tested the Great Prairie chickens at 6 different loci, and that means that probably on 6 different chromosomes, a particular area on each one. So the number of alleles went from 22 pre-bottleneck to 17 post-bottleneck. They lost 5 alleles during this time. All right, the next example is founder effect. And I'll share something with you when I get through talking about this. Okay, founder effect has to do with a small group of individuals leaving a population and establishing a new small population. This results in decreased variety of alleles carried to this new small population. Their new gene pool is different from their original population. One example of founder effect is polydactyly. Polydactyly is a symptom of a few different syndromes. Polydactyly means extra digits. These syndromes are commonly found in the Old Order Amish of Pennsylvania. Recessive genes are responsible for the traits of these disorders. In 1750s to 1850s, a small group of Amish people came to America and settled in Pennsylvania. They left Germany and other areas due to differences in religious ideas. They only brought the gene pool of those individuals. In addition, the Amish marry within their own community. So founder effect traits such as polydactyly can be seen in descendants. Well, I'll share a story with you. When I have polydactyly, and when I was 5 years old, my mother had my extra digit cut off of my hand, and I also have of both hands, and it was a type it's called an ulnar type, and it's a type B, so it wasn't a full form bone finger, and it was only like a tag of a finger-like part of just skin, okay, that resembled a small finger. And anyway, so I have been sequenced, and I have 2 variants and 2 genes that have to do with the Ellis Van Creveld syndrome. So I didn't know all this until I started working with this for you, and so that is a good day at work for me when I can put something together and learn. Okay, the next way let's see. We're going to talk about gene flow. Okay, so this is my favorite way to talk about gene flow. This is immigrants on an Atlantic liner, and they're coming to America. And so when people move into a new city, they bring their genes with them, and that's called gene flow. All right, these random processes, mutations, genetic drift, and gene flow can reduce the genetic variation of a given population and can increase the differences between populations of the same species. So our science skill today is going to be about questions and methods. And the task verb that we're going to focus on is state. And this is what we'll be stating. The null hypothesis is what we're going to be working on today, okay? And we're going to be predicting the results of an experiment. This is an FRQ from the very first year that I taught AP biology. And what you see in the diagram, you see a choice chamber. And on one side, it has a glucose-soaked cotton ball, and on the other side, it has a dry cotton ball. And so let's read what the story tells us. All right, in an investigation of fruit fly behavior, a covered choice chamber is used to test whether the spatial distribution of flies is affected by the presence of a substance placed at one end of the chamber. To test the fly's preference for glucose, 60 flies are introduced into the middle of the choice chamber at the insertion point indicated by the arrow in the figure above. A cotton ball soaked with a 10% glucose solution is placed at one end of the chamber, and a dry cotton ball with no solution is placed at the other end. The positions of the flies are observed and recorded every minute for 10 minutes. Predict the distribution of flies in the chamber after 10 minutes and justify your prediction. So take just 20 seconds and you make a prediction of where those 60 flies are going to be. All right, how about 10 more seconds? Okay, I'm going to show you my prediction. Okay, I know those are bees and they're not flies, but it's the only shape that was even resembled being close enough to put in here. Okay, so you see we have our choice chamber down the bottom, the glucose soaked, the dry, and my prediction, I predicted 58 glucose will be on the glucose soaked in, one in the middle, and one on the dry cotton. So let's take this another step, because I did that because they would want sugar, is what I was thinking. Okay, before we move on, I want to ask you a question. And my question to you, and I hope you know the answer, is what would you do differently here? What do you see as an initial problem in this diagram and the initial start of this experiment? I see that there is a dry cotton ball and there's a glucose soaked cotton ball. So I think that it would have been better to start with not a dry, but maybe a soaked cotton ball with sterile water. So it wouldn't be the wet or dry that could be interfering. It would be about the glucose. I just wanted to see if you maybe caught that on the first round. Okay, so here's B, and I'll give you a couple of minutes to do B. The experiment described above is repeated with ripe bananas at one end and unripe bananas at the other end. Once again, the positions of the flies are observed and recorded every minute for 10 minutes. The positions of the flies after 1 minute and after 10 minutes are shown in the table below. All right, I'm going to go to the next and show you the table. Okay, so here's the distribution of flies in the choice chamber at 1 minute and at 10 minutes. And there's at the end with a ripe banana. At 10 minutes, we were 45. In the middle, we had 3. And on the end, we had 12. Now, there is the chi-square formula, because you're going to be doing chi-square calculations today. You're going to write a null hypothesis, and you're going to be doing chi-square. So let's go through this formula for a minute. All right, so this symbol right here means summation, okay? And this O is talking about observed. So I would have observed 45, and on the next one it would be I observed 3, and then I observed 12. So expected, I'm going to leave that to you because you're going to be writing a null hypothesis. And so I hope that you understand what that means. And if you don't, I'll make it really clear when we get done. But one more other thing about this formula, see that chi right there, chi-square, that is not an x squared. So don't find this and then think that's x squared and then get the square root of this. Don't do that. Okay, I'm going to give you 2 minutes to work through this. And in the beginning, I should have told you to get some paper and a pen, and in the future, some graph paper and a calculator too. Okay, how about another 30 seconds? And 10. And okay, let's take a look at the answer. Okay, so to write a proper null, okay, this is what I do. And I think there's 3 things I really need to say to cover all my bases when I write this. And I always think, well, null has 3 letters, right? Okay, so I need to say that the number of flies will be equal in all 3 sections. Or I could have said it like the flies will have the same amount of preference for each area. Okay, then there is no statistical difference. And then any difference is due to chance. So I use this as a framework because I can substitute anything I'm talking about into that first sentence, and then I will be able to know what else I need to say and have some frame of reference to check myself back of, okay, what was no statistical difference with no chance or chance, but if you do it this first time when you write your null, you will be able to control which way you're writing and that you don't make an error and change things up as you're writing. So let's take a look at this math. We had 45 observed, 3, 12, and the totaling of 60. And then if we didn't expect any difference in their choices, okay, or their preferences, then there would be 20, 20, and 20. And then you would have done the calculations and come up with a chi-square value of 48.9. All right, so here's our chi-square formula, and I'm going to run through how to do the math, and then it's going to go really, really fast. Okay, I hope you were looking at the math and not trying to look at my fingers. All right, so here is with our next step. We got to take this a little bit further. This is a degree of freedom table, and this is only a small section compared to various tables. I mean, there's a lot of information can be on a table. It can be in a different orientation. The p-value can be at the top. The degrees of freedom can be on the left, the right. It can have all kind of differences, but this is the one that the College Board has on the exam. So we're in biology, we're going to use our p-value of 0.05, and now we're going to look at our degree of freedom. We had 3 categories, right? We had the ripe, we had the middle, and then we had the not ripe. So when we're determining our degree of freedom, we're going to do our number of categories that we're looking at minus 1. So this would be 2 degrees of freedom. Now our chi-square value is 48.9. So let's look and see under 2, there's no 48.9, is there? So what's happening with the numbers underneath it? So here is at 0.05, her critical value is 5.99. So we have 9.21. So what are those numbers doing? They're increasing. So 48 not 0.9 would be way down here. So as the chi-square value increases, okay, we're also going to have, let's look at our p-value. Here's p 0.05, 0.01, and as our p-value decreases, right, then our chi-square value increases. So if our p is less than 0.05, we're going to reject our null. This is not due to chance, okay? Less than 0.05, not due to chance. Okay, so this was the scoring guidelines. We predicted there was 1 point for just predicting the right number of expected in each, and then 1 point for justifying the prediction, which is our null hypothesis. Well, no, that was a separate point. The flies will be evenly distributed across the 3 different parts of the choice chambers. So that said the same thing as they preferred each equally, okay, or there would be the same number in each. And that would be because of the glucose. All right, so check for understanding. I have a multiple choice. In fact, I think I have 2 in here. And I looked up how to pronounce the, I think it's Tarika, Tarosa, or Tarisha, Tarosa, I think it's Tarika. All right, the California, let's just go with the California newt. All right, lives in the coastal areas around Los Angeles. Which of the following is a valid null hypothesis relating fitness to survival of a bottleneck event in a coastal area where a small isolated population of California newts reside? Okay, A, only those salamanders with the lowest evolutionary fitness will be eliminated by the bottleneck effect, lowering their allelic frequencies of their traits. B, Only those salamanders with the highest evolutionary fitness will survive the bottleneck event, raising their allelic frequencies of their traits. C, surviving the bottleneck event will be random, so any change in the allelic frequencies of the salamander population is not attributed to fitness. And D, surviving the bottleneck event will be random, so there will be no changes to the allelic frequencies due to the bottleneck event. Okay, I'll give you, let's go 15 seconds. Okay, I hope you chose C. Surviving the bottleneck event will be random, so any change in the allelic frequencies of the salamander population is not attributed to fitness. All right, now for some guided practice. All right, so in this table, we have some salamanders and we have their phenotype. They're a black phenotype, dark brown, light brown, and we have a flood involved, okay? There is a distribution before the flood. There's 13, 25, and 6. And then after the flood, there's 7, 14, and 3. Okay, so which of the following is an appropriate null hypothesis regarding the phenotypic frequencies of this population of salamanders before and after the flood? All right, so in A, more black salamanders survived because they could better hide from predators on the dark, muddy soil. So the frequency of the black phenotype significantly increased in the population after the flood. B, fewer light brown salamanders survived because they had reduced chance of finding mates due to the high water levels. So the frequency of the light brown phenotype in the population decreased after the flood. C, more dark brown salamanders migrated to a new, drier area because of the flood, and a fewer of them remained in their original habitat, so the frequency of the dark brown salamander significantly decreased in the population after the flood. And D, the proportions of black, dark brown, light brown salamanders before and after the flood are not significantly different, and any observed differences are due to random chance. Okay, I'll give you 15 seconds. Okay, and I hope it was practically what I told you earlier to write as a null. I hope you chose D. The proportions of black, dark brown, and light brown salamanders before and after the flood are not significantly different, and any observed differences are due to chance, to random chance. Okay, this one is so fun. All right, so you're going to need, this has to do with doing some, you're going to draw some diagrams yourself, and there's a lot of science to learn in this drawing, in this example. So let's look at our diagrams. We have an aquarium, and we have a negative control aquarium, and we have an experimental aquarium. And what else do we have? We have a male flower, we have a female flower, and we have invertebrates. Okay, let's read this story, and we're going to work on A, okay? All right, seagrasses are aquatic plants that reproduce sexually. Male seagrass flowers produce sticky pollen that is carried by circulating water to female flowers, resulting in fertilization. A researcher claims that the mobile aquatic invertebrates can also transfer pollen from male to female flowers in the absence of circulating water. To investigate this claim, the researchers set up aquariums to model the possible interactions between the invertebrates and the seagrasses. Using the symbols below and the template aquariums to demonstrate the experimental design for testing the researchers' claim, the mobile aquatic invertebrates can pollinate seagrass in the absence of circulating water. Now, draw the appropriate symbols in the negative control aquarium and the experimental aquarium, and don't use any symbol more than once in each aquarium. Okay, I'm going to give you a minute to do that. About 15 more seconds. Okay, let's take a look. All right, so look at the aquariums at the bottom. So you should have a male and a female in your negative control aquarium, and you should have a male and a female and an invertebrate in the treatment aquarium, okay? So when the purpose of the control, okay, so I think that everybody can say the control is the no treatment and the experiment is the one with the treatment. But what is the purpose of having the control? The purpose of having the control is they said that this invertebrate was responsible for carrying pollen from that male to that female. So we have to, in the negative control, see without water if it's possible to transfer pollen from that male to that female without that invertebrate variable that was added. Okay, so that was the purpose of the negative control. And that's the purpose of a control. You have to see what happens without the treatment, okay? And so you can claim that the treatment was responsible. All right, now let's do B. Identify the dependent variable in the experiment. Predict the experimental results that would support the researcher's claim that mobile aquatic invertebrates can also transfer pollen from male to female flowers in the absence of circulating water. So now that you've seen what the right answer for A is, now what is the dependent variable, and predict what the outcome is going to be of this experiment to support the researcher's claim, okay? I'll give you just a minute to do that. Okay, let's take a look at B. All right, so your dependent results should be, your dependent variable is the results, right? And that should be that fertilization occurs, okay? And what's going to happen is you should have no fertilization in the control tank and treatment and fertilization in the treatment tank. That the male is not going to be able to fertilize the female in the no treatment tank, and the male will be able to fertilize the female via the invertebrates carrying the pollen over there, okay? All right, to the female. So that should have been your dependent variable, the results, which is fertilization, and then fertilization no, and the negative control yes, and the treatment tank. So this is what the rubric looks like for this particular. And when you have one of these, if you have a prediction involved, it has to be in support of what you identified. It can't jump, like you can't have one in the third column on the bottom here supporting one in the top column over here, okay? So was I right? Fertilization was what I identified as my dependent variable. And we're going to have fertilization is seen in the experimental aquarium. I want you to also follow up with that, if you were to write that, with no fertilization is seen in the control, okay? Because we have to see if it's able to happen in the control tank without that invertebrate, okay? All right, debrief and summary. Okay, today's takeaway: evolution is also driven by random occurrences, mutations, genetic drift. Two types of genetic drift were bottleneck and founder effect. I hope you enjoyed learning about the great prairie chicken and polydactyly, and then the gene flow. I hope that you also could explain to someone else these random occurrences, and I hope you're really a whole lot better at stating a null hypothesis and predicting experimental results. Common errors and misunderstandings. A prediction must compare the experimental group to the control group. Why do we need the control? To show that a result is not occurring without the treatment. The difference between a negative control and a positive control, and my kids had in my classroom had the same question, but we had one particular lab that kind of made it all clear. In a negative control, no response is expected. No treatment is added. The dependent variable results you are looking for will not occur, okay? In a positive control, will reflect a known result. And a good example of this is when you're running gel electrophoresis. A positive control will tell you if a result is in fact negative because it's really negative and not just due to operator error. If the positive result was missing, then we know it's not really a negative result, but something else in your procedure probably went wrong. All right, we have some homework left. All right, so tonight, and this is another one of my favorite questions. In fact, I start my kids off at their very first day of school every year on day one. We do this. We learn how to graph, and we learn how to place error bars and just get that right from day one. So you have a table, you have bees involved in this, okay? And you have caffeine, and you're going to look at putting error bars, plus or minus 2, standard error, the mean, and you have 10 minutes and 24 hours, okay, that they had an average probability. All right, so in flowering plants, pollination is a process that leads to fertilization of an egg and the production of seeds. Some flowers attract pollinators, such as bees, usual visual and chemical cues. When a bee visits a flower, in addition to transferring pollen, the bee can take nectar from the flower and use it to make honey for the colony. Nectar contains sugar, but certain plants also produce caffeine in the nectar. Caffeine is a bitter tasting compound that can be toxic to insects at high concentrations. To investigate the role of caffeine in nectar, the group of researchers studied the effect of 0.1 millimole caffeine on bee behavior. The results of an experiment to test the effect of caffeine on bees' memory of a nectar source are shown in Table 1. Design an experiment using artificial flowers to investigate potential negative effects of increasing caffeine concentrations in nectar on the number of floral visits by the bees. I'm going to give you the road to not go down, okay? I don't want you to talk about the bees overdosing on caffeine, okay? I don't want them dying. I just want you to increase those caffeine concentrations and have a nice experimental design. All right. Identify the null hypothesis, an appropriate control treatment, and the predicted results that could be used to reject the null hypothesis. Now, I added this to this because we've got to get this skill down. I want you to make a graph also, okay? And tomorrow night when the next lesson when Ms. Kate starts her lesson, it will start off with the answer to this homework, okay? Enjoy that question. All right, on device and internet access, we know that not all students have access to the internet or a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know, cb.org/tech. And I want to give a shout out to Margaret from Alaska and to Mila. Thank you for your questions this week. Thanks for watching. We'll hope you'll join us again. Be sure to complete the practice question and we'll review the answer in the next lesson.